Everybody, in this video we are going to talk about a new or another DNA repair process. This one is called the mismatch repair. So to put this in some context with the other repair pathways, or some of the other repair pathways we have talked about, let's take a look at them briefly. So we have base excision repair, which is predominantly in charge of fixing things like when you have uracil inside DNA. So uracil here we have paired with G, that's not normal, so base excision repair would take care of that one. Here's thymine glycol, which is a modified base, which should not be in DNA, and base excision repair can take care of that. And here's another example, 8-oxo-G. Now, 8-oxo-G is a modified base that should not be in DNA, and base excision repair can take care of that one. We've also looked at nucleotide excision repair. So nucleotide excision repair, NER, fixes cyclobutane pyrimidine dimers and other types of damage that cause what we will call structural defects, say in the double helical structure of DNA. So nucleotide excision repair will fix those types of DNA lesions. Excuse me. So and today, or in this video, we are going to look at mismatch repair, MMR. And this repair process is in charge of normal mismatches. So mismatches between just normal nucleotides. So here we have a T paired with a G. Now C should be paired with G, not T. So mismatch repair would fix a mismatch like this. Also, let's see, C with A. Okay, so that's not a normal pairing, and mismatch repair would fix a mismatch like this. Mismatch repair also fixes small indels, and I think this is the first time we use this word in this course. So indels, that's a jargon term for insertion or deletion in DNA. And as indels, what they can do is they can cause loops, small loops to occur inside the DNA molecule. And I can show you how an indel can lead to a small loop in the DNA in a moment. Let me see, how did I want to do that? Okay, in a little bit I'll talk more about how indels can be repaired by mismatch repair. So another thing we need to address when talking about mismatch repair is this process is in charge of microsatellite stability. So what are microsatellites? Now, we've seen what satellites are earlier in the course, right? Satellite DNA, that's the DNA that was not migrating at the same density level with most of the genomic DNA during density gradient centrifugation assays. And researchers found that satellite DNA is usually made up of repetitive DNA. So I'm not exactly sure on the history of this term, but my guess is microsatellite, this term came up from the fact that microsatellites, our current definition is that these are repetitive DNA, but a certain type of repetitive DNA. For example, a microsatellite is defined as a repeating unit of nucleotides. Now, specifically, 2 to 10 nucleotides. I guess this is the common range of a microsatellite. So 2 to 10 nucleotides repeated 5 to 50 times. So this is the definition we'll use for a microsatellite. And maybe, occasionally we go out of this range. Maybe there'll be 11 or 12 nucleotides or slightly more. And maybe the repeats will be slightly higher than 50 times. But for the most part, microsatellites are defined as this range. And what do we mean by 2 to 10 nucleotides? So let's just go with 2. So if we had a locus somewhere in, say, the human genome, that had this sequence, GA, followed by another GA, and again, and again, and again, 1, 2, 3, 4, 5. Okay, this would be considered a microsatellite locus. So we have 5 repeating units of GA. Now the other strand would be CT, CT, right? Like this. And you can imagine that, we could expand this to, according to this definition, up to 50, and that would count as a microsatellite locus. Now, microsatellites can shrink, shorten, and expand. And the mechanism behind shrinking and expanding is thought to be, what's the word I'm going to use? Replication slippage. Replication slippage. Now, for the most part, microsatellite loci are relatively stable, and as a result, they are commonly used for DNA fingerprinting. Now, I'm not going to spend too much time on DNA fingerprinting. Actually, I'm not going to talk any more about DNA fingerprinting except for mentioning it here, but they are used for DNA fingerprinting. And I encourage you to take a look at that. You can look up some information on that if you want to know more about it. But so they're relatively stable, unless mismatch repair is not working. But when changes do occur to microsatellites, it's thought to be due to this mechanism called one of the primary mechanisms leading to changes in microsatellites shrinking or expanding is thought to be replication slippage. Now, I'm going to cheat a little bit here. I have some diagrams made up for you. So let's say this is a microsatellite locus and we have CTs repeated on the top strand and GAs repeated on the complementary strand. So when this is replicated, right, what happens? These strands need to be separated. And let's say we're just looking at synthesis of the CT strand using the GA strand as the template. So the other one's going to be copied too, but I just want to focus in on replication with the GA strand as the template. Now the polymerase will start replicating this, and say most of the time everything works out just fine. The polymerase replicates this and it doesn't have any trouble. But if something should go wrong, and let's say the polymerase should dissociate from the template, and let's say down here what we have is dissociated from the template and the strand separated. Now, for replication to continue, the polymerase will have to be loaded back onto the template, and then something will need to happen to this thing right here. Now, it could re-anneal perfectly with this template down here, or it could come back in a way where it makes a little loop. So you can see right here, these guys should have annealed. Ideally, they would have annealed down here with these G's and A's. But they didn't, and it re-annealed with a little loop right here. So if the polymerase should start replicating from here, the result is going to be that the top strand is going to have a little loop, extra loop of DNA. It's going to be a little longer, than that bottom strand. And we could say right here, this one is 1, 2, 3, 4, 5. This is a 5 nucleotide loop. Now, mismatch repair is in charge of searching the DNA for these little loops and removing them. Now, the same thing can happen with the other strand, right? So here I've diagrammed it. So let's say that a polymerase fell off of the template up here. This is the template. It fell off while it was replicating this one, and when the strands came back together, when the polymerase was loaded back on the template and the strands came back together, it came back in such a way that we have a little loop in the template strand. So after this has been replicated, what we have, after replication finishes, what we'll have is a little loop down here. So in these opposite cases, we have sort of, if this is not repaired, if this top strand here is not repaired before the DNA undergoes replication again, what we'll have is an increase, right, in the total length of the microsatellite repeat with respect to this top strand, right? And down here, what we would have is a decrease. So that's assuming DNA replication occurs, again, on these completed molecules before these little loops have been repaired or properly repaired. And we're going to talk in a moment about determining which one is, how does the cell know which one should be repaired? Should the loop be removed, or should more bases be added down here? We're going to talk about that in a moment. So again, replication slippage can lead to increasing the length of the microsatellite, increasing the number of repeats in the microsatellite, or decreasing the number of repeats. And it depends how these loops come back together, and it depends on another round of replication occurring before the loops are repaired. Okay, I hope that wasn't too confusing. Now, okay, so let's say mismatch repair is not working. We're going to see a genetic disorder that results when mismatch repair is not working towards the end of the lecture. But one of the things that happens is the microsatellite loci become unstable. Microsatellite loci, unstable. And this would be in the human genome. We'll use humans as an example. And we can also expect an increase in mutation rate just throughout the genome. Increase in mutation rate relative to normal. Okay, that's what happens when mismatch repair is not working. So, how does mismatch repair work? So, before we get into that, there is a dilemma. We have a dilemma, right? So, what is the mismatch repair dilemma? So, let's take a random sequence of DNA here. And I wanted to put a mismatch. I'm going to put a mismatch right here, this G with a T. And, I could put some bases in here too. So here we have a mismatch. Now, the dilemma is which one is incorrect. Is it the G that should be changed to an A, or is it the T that should be changed to a C? Which one is incorrect? So if mismatch repair just chose one at random, if they chose randomly, it would be wrong at least 50% of the time. And sometimes mismatch repair needs to choose randomly, but there appears to have be mechanisms that help mismatch repair determine which one should be repaired. Now, in both prokaryotes and eukaryotes, it seems that mismatch repair has evolved to, change the base in the most recently synthesized strand. And the hypothesis, or the common thought behind this, is that the errors are most likely to be due to replication of the new strand, and thus the template is less likely to have the errors than the newly synthesized strand. So DNA polymerase can make mistakes when it's replicating, and it is more likely to make an error making the new DNA than there is to be an error in the template itself. So mismatch repair seems to have evolved to try to bias replication, or bias fixing the DNA to changing the sequence of the most recently synthesized strand, as opposed to the strand that was used as the template. Now, the mechanism used by prokaryotes and eukaryotes to determine which is the new strand, which strand is most likely to be the new strand, is different. So both prokaryotes and eukaryotes have mismatch repair. And we understand very well how the process works in prokaryotes. We don't really understand it that well in eukaryotes. So in prokaryotes, the dilemma appears to have been solved by using the methylation status of the DNA. And this doesn't really work in eukaryotes, because eukaryotes, even though eukaryotes use DNA methylation, the genome isn't uniformly methylated, as we will see, as it is in prokaryotes, and I'm gonna explain that in a moment, or at least E. coli. So the mechanism used by eukaryotes to distinguish the new strand from the old strand is unclear, but it may involve DNA nicks. And if it does involve DNA nicks, the common thought is that it's because of the Okazaki fragments. Okazaki fragments, right? Which, during DNA replication, will have some nicks associated with lagging strand synthesis and the Okazaki fragments. However, this model is not perfect, and there's still many questions as to how eukaryotes distinguish the new strand, or the more recently synthesized strand, from the template strand. But let's take a look at the prokaryotic method. So prokaryotes, in order to choose the right strand, prokaryotes use the methylation status of their DNA, and there's a key enzyme that is in charge of methylating the genome. It's called DAM, for deoxyadenosine methylase. And so what this enzyme does is it methylates adenine, and it methylates it in a specific way that the cell doesn't mind. So base excision repair is not going to remove these methylated adenines like it might remove certain methylated forms of adenine that were, let's say, methylated in a different way. So let's say, let me show you, I'm going to show you with a diagram of adenine right here how DAM methylates. There's an N here, how DAM methylates adenine. So if this is a structure of adenine right here, I think I have it right, I'm missing a double bond here. This is adenine. So DAM here is going to methylate adenine in a way to make 6-methyl adenine. So it's going to look like this. And maybe if some of you remember the numbering pattern for purine rings here, you might know where that 6 is going to go, where the 6th position is. Oh, there's an N in there. I always forget that N. So we're going to put it right here. Methyl group, so that carbon is linked to the nitrogen here. This is where the glycosidic bond forms with the 5-carbon sugar. So this is 6-methyl adenine right here, and this is regular old adenine. And now base excision repair will not remove this. This is perfectly normal for DNA in prokaryotes, like E. coli. Now, so DAM doesn't methylate every adenine in the genome. It methylates just the adenines in this sequence right here. So we have 5' GATC, there's one A in there, it's going to methylate that one. In the complement here, it's going to be the same sequence, right? It's like a palindrome here. CTAG. So GATC this way, GATC that way. Now this A is going to be methylated too. Okay, so everywhere we see a GATC, or it sees a GATC in E. coli genome, the A will be methylated. So let's take a look at a part of the E. coli genome here. Now I'm going to put in some GATCs in here so you can see how this relates to mismatch repair. GATCTT. I'll put some A's in here. Okay, put the complement in here. A, G, A, T, C, G, C, G, T, G, A, T, C, A, T, T, G, A, T, C, T. Okay, so let's find all the GATCs. Methylated. Methylated, okay. Methylated. Okay, now, when this is replicated, let's just take a look at the bottom strand here. We'll ignore the top strand. So the bottom strand is the template. I'm going to put these guys in here. You can kind of guess, maybe start to guess how mismatch repair is going to use methylation to distinguish the two strands. While I fill these in, let me use a different color. Why not for the new? Okay, so polymerase comes in and it's replicating from the 5 in the 5' to 3' direction, and it's putting in regular adenines here, so they're not methylated yet. Ooh, I forgot to put these in here, right? So this is the template. So template's methylated, and as polymerase comes in here, DNA polymerase, it's replicating these. It's using regular adenines, adenine nucleotides. Okay, so at this point, the DNA is hemimethylated. So only half of it is methylated, right? Hemimethylated. So I can change this. Why don't I put a G here? Okay, let's say there's a mismatch here. There's a G mismatch with T. Now, DAM comes in here and methylates at some point after replication, but not right after, not immediately after replication. It doesn't like follow along with the polymerase immediately methylating. So at this point, mismatch repair proteins can find the mismatch and look at the methylation status of the two strands, and they can see that this one down here is methylated, this one up here is not methylated. So this is the mismatch, and this is the one that needs to be fixed. Okay, so that's how the process works in prokaryotes, the process of distinguishing the two strands and determining which base is the one that needs to be fixed. And unlike base excision repair, well, even certain aspects of base excision repair involved what the short path and the long path pathway, replacing more than one base. But with respect to mismatch repair, we're going to replace more than one base. We're going to replace a big segment here of the DNA, or the process mismatch repair is going to replace a relatively long segment of the DNA when it's replacing the mismatch. When it's replacing the mismatch, it's going to replace a segment of the DNA, not just the mismatched base. Okay, so let's take a look at how the mechanism works in eukaryotes. Like we said before, we're not exactly sure how eukaryotes distinguish the new strand and the old strand. So MMR mechanism in eukaryotes, but we know a lot of the proteins involved in the process of fixing the match. And we know nicks are important. Nicks in the DNA seem to be important. And these studies are mostly from in vitro repair assays, where researchers have figured out a lot of the proteins involved in mismatch repair, and they can put them in a test tube and put nicks on certain strands of DNA and watch certain the mismatch be repaired in certain ways depending on where the nick is located in the DNA molecule. So in vitro assays have been key to helping us kind of understand the mismatch repair mechanism in eukaryotes. So let's take a look at some of the protein complexes. So there are two primary complexes that are important. So mut-S, the A mut-S complex, and A mut-L complex. And there are a couple different of these versions of these complexes. So mut-S alpha and mut-S beta for the mut-S complexes. So alpha is made up of two proteins, MSH2 and MSH6. Beta is made up of MSH2 and MSH3. Now, the alpha complex seems to be important for regular mismatches and loops smaller than 5 nucleotides. Beta seems to be important for loops bigger than 5 nucleotides. Bigger than 4 nucleotides, let's say. So beta is in charge, mut-S beta is in charge of loops that are 5 nucleotides and longer. So mut-L, and I should say the mut-S complexes are in charge of detecting the mismatches and problems with the DNA. Mut-L, this is more of a endonuclease. Or I should say our current models, or at least the models that from the references I used to put together this lecture, these mut-L complexes have the endonuclease activities, which are going to be important for mismatch repair. So we have 3 versions: mut-L alpha, mut-L beta, mut-L gamma. So alpha is a complex of MLH1 and PMS2, so 2 proteins there. Beta also has MLH1, but instead of PMS2, it uses PMS1. And gamma also uses MLH1, and the second protein in that one is MLH3. So you can see with both the mut-S complexes, there's one key protein. MSH2 is in both of them. And for mut-L, MLH1 is in all 3. So I've seen some references refer to these like MSH2 as the scaffolding protein, and MSH6 or MSH3, these are specificity factors. MLH1, in that case, MLH1 would be the scaffolding protein, and these other guys here would be specificity factors, which kind of give unique properties to each of these complexes. However, we still don't know too much about how these mut-Ls differ from one another. So what we're gonna do for this course is just focus in on the mut-S's important for detection, the mut-L's important for their endonuclease activities. And I'm going to diagram the mechanism for you, and we're just gonna focus in on mut-S alpha and mut-L alpha. We won't worry too much about these others here. So how does the mechanism work? Well, the model we're working with goes like this. Okay, so here's a mismatch in a DNA molecule. Now, the cell needs to decide which one it's going to fix, and it's going to focus in here on this nick. We have a nick in this strand of DNA here. So as a result, because this nick is here, this is the one that's going to be fixed, and it's going to be replaced with an A. And again, we know this from in vitro studies. If the nick was placed up here in the in vitro experiment, then it would be the T that is replaced. But because the nick is down here, it's the G that's going to be replaced. So mut S alpha comes in first. It's the one that recognizes the mismatch. So here I have it diagrammed already for us. Mut S alpha is here. It recognizes the mismatch. Then mut L alpha is going to come in. And make sure I have these in the right order. Okay, so now that mut L alpha is here, so mut S alpha recruits mut L alpha to the mismatch. And at this point, replication factor C and PCNA come in, and these associate with the nick. And once these are on here, these guys all communicate. PCNA, RFC, mut S alpha, mut L alpha, and this DNA probably loops around. I have it drawn like flat, just for going over the mechanism, but it probably loops, and these guys might all interact and connect. Again, that's a part of the model I'm not that sure on. I'm not sure researchers are that sure on it yet either. So, but at this point, mut L alpha will nick this strand down here. It will nick the strand that has already been nicked, and it will nick the strand on the opposite side of the mismatch, the opposite side relative to the prior nick that was already in the DNA. So it's going to place a nick here. So once we have a nick here, I wonder if I should put a little nick in there. Yeah, why not? So OH here, phosphate here. Now, exonuclease comes in. I think we know that exonuclease is called exonuclease 1, but we can just think of it as we don't have to know the specific exonuclease, but exonuclease comes in and it removes the DNA between the two nicks. So we have something like this, mut L alpha, mut S alpha. We have the T here, that G has been removed. Then we have this long gap here. So now that's when DNA polymerase comes in. Well, which way are we going? We're going this way. We're going this way. We're going to synthesize using the top strand here with the correct base as the template. And then this is the 5' phosphate. DNA polymerase will leave a nick that needs to be repaired by DNA ligase. And okay, so that's it. That's it for the mismatch repair mechanism. Again, we're still, I guess as far as a few years ago at least, I haven't had time to check the most recent research on this mechanism. We're still unsure where those initial nicks come from, but it might have something to do with Okazaki fragments during replication. But for the most part, that's the mismatch repair mechanism, so we don't have to worry about more details than what I just covered in this video. Now, I want to talk about one disorder related to mismatch repair that is called Lynch syndrome. So Lynch syndrome is essentially, I guess another term for it is hereditary non-polyposis colorectal, I don't know how to spell that, colorectal cancer. I think there's only one R here. So abbreviated HNPCC. So essentially it's hereditary colon cancer, and it is inherited as a autosomal dominant trait. Autosomal dominant, what does that mean? It means the genes that are correlated with the syndrome are found on non-sex chromosomes. So, and one of the hallmarks of Lynch syndrome is the tumors nearly all tumors show high microsatellite instability. So high microsatellite instability, high MSI, and this is often abbreviated like this, MSIH. So looking at the tumors, looking at the microsatellite loci, they're growing, they're shrinking, they're just all tons of mutations in the microsatellite loci. And when researchers investigated this, they found out, what did they find out? Is that, okay, so what are the genes involved? Well, MSH2 is mutated in 60%. So both copies of MSH2 are mutated in 60% of patients with Lynch syndrome. That's a mismatch repair protein, right? It's part of MUT-S alpha and beta. MLH1, both alleles of MLH1 are mutated in 30% of Lynch syndrome cases. So this is the protein involved in all of the MUT-L complexes. MSH6, which was in the specificity factor in MUT-S alpha, in the MUT-S alpha complex, is found in 7 to 10% of patients with Lynch syndrome. Not found, but both copies are mutated in 7 to 10% of patients with Lynch syndrome. So then we have other genes, PMS2, PMS1, MLH3. So these were all part of the MUT-Ls, various MUT-L complexes, and these are each found in less than 1%. So why? Well, we already learned that mismatch repair is important for microsatellite stability. And when we lose mismatch repair, well, then the microsatellites tend to get lots of mutations during replication, probably by replication slippage. And those little loops go unrepaired, and leading to the microsatellite instability. So what is this autosomal dominant? So address the autosomal dominant nature of these mutations. And what do we mean by autosomal dominant with respect to these mutations? What we mean is that inheriting just one mutant allele, whether it be MSH2 minus, and let's say the other one is MSH2 plus. So say a human inherits MSH2 minus from one parent and MSH2 plus from the other parent. Inheriting one non-functional allele is sufficient to predispose one to cancer. So that's what we mean by dominant. Even though this human has a functional MSH2, inheriting just one non-functional copy predisposes the human to cancer.